it is the time to discuss fungus-like organisms with uncertain affinity. These members are now considered as protists, although earlier these were called slime molds and traditionally studied by the mycologist, sometimes plant pathologist also. Now the phylum Myxomycota. Somatic body of Myxomycota is multinucleate protoplast, wall-less and without any definite form, and this protoplasm exhibit reversible subtle streaming. Now, somatic body of Myxomycota are diploid, holocarpic and free-living. In the slide, you can see the diploid plasmodium of Myxomycota. Now, three different basic types of somatic body or plasmodia are characterized under Myxomycota, of which one is called protoplasmodium. These are very minute, microscopic throughout their existence. Second type is aphanoplasmodium, which is hyaline and produce thread-like network. However, this hyaline nature is mainly due to agranular cytoplasm. The third type is phaneroplasmodium, which is conspicuous, prominent and visible due to their granular cytoplasm. Now, three different types of uninucleate cells are observed in the life cycle of Myxomycota, of which at least one is flagellated structure. Now, Myxomycetes exhibit two different types of nuclear division. One is centric or open type, where no persistent nuclear envelope is present, which breakdowns in the prophase, and during cell division or nuclear division, centriole remain present in association with the spindle apparatus. In acentric type, nuclear membrane usually remain intact until late anaphase or late telophase. Now, during asexual reproduction, stationary sporophores are produced, and these sporophores are very much like that of other fungi. Actually, during asexual reproduction, the plasmodial mass aggregated to form a pseudoplasmodium, from which initially a stalk-like structure appears, at the tip of which a swollen sac-like spore-containing region is present. This spore-containing region remain covered by a non-cellular periderm, as you can see in the diagram. Inside the peridium, the capillitium along with the spores remain confined. The spores on germination gives rise to amoebae. During sexual reproduction, usually two swarmer cells behave like gamete. They fuse with each other, form a typical dikaryotic structure, which in turn after karyogamy gives rise to zygote. And from zygote on germination, the phaneroplasmodium or the dominant, prominent, visible plasmodium are produced. Resistant stage of the Myxomycota are represented by the sclerotia. If you cut a section of sclerotia, what you will find? You will find large number of macrocysts remain scattered inside it. Actually, these Myxomycetes members exhibit phagocytosis. They engulf bacteria or other objects, larger objects, as their food. The next group is the Plasmodiophoromycota. Members of these Plasmodiophoromycota are usually necrotrophic, biotrophic parasite. The members always induce hypertrophy and hyperplasia within the host cell. After infection, they induce the surrounding cells around infection coat to divide first, so a hypertrophic condition is produced, which is the typical symptom produced by the members of Plasmodiophoromycota in cabbage or other brassicaceous plant. This slide shows the symptom produced by the Plasmodiophoromycota, and this is the hypertrophied and hyperplastic condition of the cells. The somatic cell of Plasmodiophoromycota is multinucleate plasmodium, which exhibit amoeboid movement when only inside the cell, but they never exhibit reversible settling as in case of Myxomycota, or they are unable to phagocytize their food material as such. Now, in the life cycle of Plasmodiophoromycota, two different types of plasmodium is observed. One is called primary plasmodium or sporogenic plasmodium, which is haploid in nature and responsible for production of zoosporangium and zoospore. Now, in the diagram you can see that after liberation of zoospore from the amoebae, the secondary zoospore or primary zoospore infect the root cells, and when inside the cell, their nucleus divide and redivide to give rise to a multinucleate plasmodium. This plasmodium is actually referred to as primary plasmodium or sporogenic plasmodium, from which the secondary zoospores are produced. Since these biflagellated zoospores are produced from this type of primary multinucleate plasmodium, not from the resting spore, these are called secondary zoospore. In the second phase of life cycle, a diploid secondary plasmodium is produced, which is also known as cystogenous plasmodium, because it is responsible for production of resting spores mainly. Now, nuclear division in this particular group is called cruciform type nuclear division. During nuclear division, the nucleolus elongates and the entire chromosome form a ring around the elongated nucleus, and after separation of chromosomes into the poles, two groups of chromosomes remain present. If you view the entire cell from a side, you will find it in form of a cross, and that's why it is known as cruciform division. Asexual reproduction in this group takes place by means of biflagellated zoospores, which after their resorption of flagella encysted and infect the host cell by a special mechanism. I am not going to describe the mechanism of infection here. During sexual reproduction, usually the secondary zoospores behave as gamete, they fuses with each other and form a dikaryotic quadriflagellate structure. That quadriflagellate structure, now you can see in the diagram, infect the root hair cells, and inside which the diploid nucleus undergoes division and redivision to form a multinucleate protoplasm, or you can say plasmodium, which is called cystogenous plasmodium and diploid in nature, from which the resting spores are differentiated. But according to some other mycologists, that sexual reproduction very rarely occur in this group. So, just to follow the diagram, that zoospores after liberation infect the host cell and eventually transformed inside the host cell into a diploid structure from which the cystogenous or diploid or secondary plasmodium differentiates. Within the secondary plasmodium or cystogenous plasmodium, the resting spore with thick walls are produced, which after degradation of the host tissue liberated into the environment germinates and gives rise to the primary zoospore. Now comes to the members of Dictyosteliomycota. In this group, the somatic body is represented by a unicellular, small, inconspicuous haploid amoebae, which are typically provided with Plasmodia, pseudopodia, which are very fine thread-like structure produced for their movement. Now, in some cases, these individual amoebae aggregated to form pseudoplasmodium with typical radiating arms, as you can see in the diagram. Now, this structure is nothing but somatic aggregation of amoebae. This is technically called as grex or slug. During asexual reproduction, what happens? From the slug, a small, short, stalk-like structure is produced carrying a swollen cap-like structure or sac-like structure, and the entire structure is usually called as sorocarp. Inside the sorocarp or the swollen tip, the spores are aligned or present with their typical cell wall. So, the sorocarp produces with walled spores and are never covered by peridium as observed in the Myxomycota. Inside the sorocarp, no capillitium is observed. During asexual reproduction, the amoebae undergoes mitotic division to form two daughter nuclei, and ultimately this leads to increase in number of the somatic amoebae within a colony. Now, another type of asexual reproduction is the formation of microcyst, and it is formed under unfavorable conditions. During unfavorable condition, what happens? The individual amoebae encysted and gives rise to what we called the microcyst. With the onset of favorable condition, this microcyst germinates and gives rise to the amoebae proper. During sexual reproduction, dictyostelids fuses their two individual amoebae, which are called giant cell, and this giant cell is nothing but a dikaryotic structure. This dikaryotic giant cell enlarges in size, attract unfused amoebae around it, and then produce a primary wall or sheath as such. After formation of primary wall, what happens? The giant dikaryotic cell or the zygote voraciously eat the unfused amoebae for their growth and nutrition. And finally, a solid wall is produced around to give rise to what we call macrocyst. Now, this macrocyst germinates to produce haploid amoebae, but karyogamy and meiosis in this particular organism is very much delayed, and meiosis occurs just prior to differentiation of spores. So, haploid amoebae are produced from the macrocyst. Now, the next group of organism, that is Acrasiomycota. Unlike Dictyostelids, the somatic body of the acrasids are cylindrical rather than flattened in outline. They are provided with a large anterior lobose pseudopodia, and they exhibit forward burst of the cytoplasm during their movement. However, the posterior end of the plasmodium is known as uroid, which is basically a knob-like structure with phylous pseudopodia and provided with a large contractile vacuole. This is the structure of uroid, and inside a contractile vacuole is present, and this black arrow denote the movement of amoebae. Now, the feeding behavior of acrasids. Actually, acrasids are phagotrophic in their nutrition, so they feed on yeast, bacteria, or other unicellular similar members. Sometimes these acrasids can engulf rather big objects during their feeding. Now, in the slide, you can show the series of pictures how acrasid phagotrophically feed on the bacteria. Mitochondria of this group of organism is with plate-like or disc-like cristae, and its nucleus is typical as in other organisms with suitable typical centrally placed nucleoli. And in this picture you can see mitochondria with small plate-like, which appears white, disc-like cristae. During unfavorable condition, the amoebae insisted to form microcyst. At the outset, they form a small body around which they secrete a wall and convert it into microcyst. During germination, the wall of the microcyst cracks, and through which the amoebae are liberated, as you can see. So, the conversion between flagellate, amoebae and microcyst occur in the nature regularly. Under favorable condition, the amoebae of acrasid gathered together, they form a pseudoplasmodium, and within the pseudoplasmodium, entity of the individual amoebae is never lost. During this period, they form a very small microscopic tree-like structure with a small stalk and the branches. So, within the branch, you can see in the diagram that individual amoebae align themselves in a single row. They encysted and converted into spore. After their dispersal, they germinate to give rise to the cylindrical somatic inconspicuous small amoebae. However, the amoebae which constitute the stalk-like portion of the branched tree-like saurocarp also germinate and give rise to the amoebae as such. Sexual reproduction is completely unknown in this group. So, this is about the characteristic features of Myxomycota, Plasmodiophoromycota, Dictyosteliomycota and Acrasiomycota we have discussed in this resume. Now, it is the time to compare the two classificatory systems I have already discussed, one proposed by Ainsworth and another proposed by Deacon in his book Fungal Biology in the year 2006. 6. Now, you see that Ainsworth divided fungi into two divisions, Myxomycota and division Eumycota, but Decon placed the members of Myxomycota, giving a status of just phylum under the group of fungus-like organism with uncertain affinity. So, this is the difference between a natural system of classification and a mere phylogenetic system of classification. Now, the true fungi were placed on the division Eumycota by Ainsworth, which were divided into 5 different subdivisions: Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, and Deuteromycotina. In this system of classification, Deuteromycotina is completely abolished, and the members of Basidiomycotina and Ascomycotina, or the hyphal members or filamentous members of these two groups of fungi, which reproduce absolutely by means of mitospore or asexual spores, never placed in this group. Members of Zygomycotina segregated into two, one Zygomycota and another is Glomeromycota. The reasons behind creation of the phylum Glomeromycota by Decon is explained already. The members of Basidiomycotina and Deuteromycotina were kept as such, but different groups recognized by Decon or contemporary mycologists is quite different, as you have noticed earlier. Now, the other members like Plasmodiophoromycota, Hydromyxomycota, and Myxomycetes, Acrasiomycetes are placed under the head fungi-like organisms with uncertain affinity. This is true that even analysis of 18S RNA, or rather you can say 18S rDNA, failed to solve the puzzling problem regarding their placement under suitable taxa. So that's all. Thank you.